Good morning. Welcome back to Daily Lectures by New Age Cosmetic White Edtech Healthcare Consultancy. I'm Dr. Bobby, and we are on the subject of molecular biotechnology. And we are on the topic of vaccines and the subtopic bacteria as antigen delivery systems. Antigens that are located on the outer surface of a bacterial cell are more likely to be immunogenic than are those in the cytoplasm. This one strategy is to place a neutralizing antigen from a pathogenic bacterium on the surface of a live, non-pathogenic bacterium. Flagella are made up of filaments of a single protein called flagella. Under a microscope, they appear as conspicuous thread-like structures on the outer surfaces of bacteria. If the flagella of a non-pathogenic organism could be made to carry a specific epitope from a pathogenic bacterium, protective immunogenicity might be easily achieved. This strategy was used to engineer a cholera vaccine, a synthetic oligonucleotide specifying an epitope of the cholera toxin B subunit was inserted into a portion of the Salmonella flagellum gene that varies considerably from one strain to another hypervariable segment. The construct was then introduced into a flagellum-negative strain of Salmonella, the epitope which consisted of amino acid residues 50 to 64 of the cholera toxins B subunit was shown previously to elicit antibodies and directed against intact cholera toxin. The chimeric flagellum functioned normally. Furthermore, the epitope was present at the flagellum surface. Immunization of mice by intraperitoneal injections of approximately 510 raised to 6 live or formalin-killed flagellum engineered bacteria elicited high levels of antibodies directed against both the peptide, that is amino acids, 50 to 64, and the intact cholera against both the peptide, that is amino acids, 50 to 64, and the intact cholera toxin molecule. 2 or 3 different epitopes can be inserted into a single Salmonella flagellum gene by creating a multivalent bacterial vaccine. Attenuated Salmonella strains can be administered orally, which would enable them to deliver a range of bacterial, viral, and parasite antigens to the mucosal immune system. For this purpose, the choice of the promoter that drives the transcription of the foreign antigen is important. If too strong a promoter is used, the metabolic load might constrain bacterial proliferation. Moreover, unlike a closed system such as fermentation vessel, shifting the temperature or adding specific metabolites to induce foreign gene expression is not possible when the bacterial vector is added to a host animal. On the other hand, promoters that respond to environmental signals may provide effective means of controlling the expression of the foreign antigen gene. For example, the E. coli NIRB promoter, which is regulated by both nitrite and oxygen tension of the environment, becomes active under anaerobic conditions. In one series of experiments, the NIRB promoter was used to direct the expression of the non-toxic immunogenic fragment C of C. tetani toxin, tetanus toxin, in an attenuated strain of Salmonella. It is estimated that more than 1 million deaths per year in the developing world are the result of C. tetani infections. When the engineered Salmonella strain was grown aerobically in culture, tetanus toxin fragment C was not synthesized. However, following oral administration of the bacterium to test mice, fragment C was produced and the animals generated antibodies against the peptide. Thus, the engineered Salmonella strain has potential as a live oral tetanus vaccine. The spiral-shaped gastrointestinal and microaerophilic gram-negative bacteria, Helicobacter pylori, is widely distributed among human populations. It is believed to be the causative agent for a number of gastrointestinal diseases, including chronic gastritis, peptic ulcers, gastric lymphoma, and gastric cancer. Among infected individuals, which includes more than half of the world's population, about 10% are at risk of developing peptic ulcers. In recent years, the medical treatment of peptic ulcers has changed from antacids to antibiotics and proton pump inhibitors. The antibiotics eradicate the H. pylori infection, while the protein pump inhibitors block the enzyme hydrogen potassium ADPase, preventing the production of acid from the parietal cells at the gastric mucosa, which facilitates the healing of the mucosa. Unfortunately, H. pylori is resistant to a number of commonly used antibiotics, and hence, including metronidazole, amoxicillin, erythromycin, and clarithromycin, seem to have less of an effect. So treatment of H. pylori requires multi-drug regimens because the organism resides in a layer of mucus that acts as a barrier to antibiotic penetration. In addition, the necessary course of antibiotic treatment is too expensive for populations of less developed countries. Colonization of the gastrointestinal tract by H. pylori is facilitated by the action of an H. pylori encoded urease. This enzyme hydrolyzes urea to carbon dioxide, and ammonia, thereby neutralizing stomach acid, making it possible for the bacterium to survive, bind, and function in the host. Urease is a cytosolic and surface-exposed nickel metalloenzyme and is one of the most abundant expressed proteins in H. pylori. The enzyme comprises two subunits, A and B, that assemble into a complex alpha B3-4 supramolecular structure. Subunit B is more antigenic, making it possible to be a vaccine candidate. To develop a vaccine that protects individuals against H. pylori infections, the genes encoding H. pylori urease subunits A and B were constitutively expressed under the control of a salmonella promoter and a genetically deleted, attenuated strain of S. enterica syrova typhi. Neither immunization with urease expressing S. enterica syrova typhi alone, nor immunization with purified urease enzyme, plus an adjuvant confer protection against challenge with a mouse-adapted strain of H. pylori. On the other hand, a vaccination protocol that combined both urease expressing S. enterica syrova typhi and urease plus an adjuvant was protective. While the success of this approach remains to be established in humans, these initial results are nevertheless encouraging and giving hope that a human vaccine against H. pylori will be developed in the near future. I think it's already through in the present day events. I'll just update info on that and give you a feedback. Okay, the summary on vaccines. Traditionally, vaccines have been either inactivated or attenuated infectious agents, bacteria, viruses, that are injected into an antibody-producing organism to produce immunity. There are a number of drawbacks to these vaccines. For example, not all pathogenic organisms can be grown to large volumes needed to make a vaccine. There are safety concerns when large volumes of pathogenic organisms are being handled. Attenuated strains may revert to the infectious state. Inactivation may be incomplete, and shelf life is often dependent on refrigeration. Recombinant DNA technology has been used in various ways to create reliable vaccines. Immunologically active, non-infectious agents are produced by deleting the genes that cause virulence. With this deletion, a live vaccine would never be able to revert to the infectious form. A gene that encodes the major antigenic determinants from a pathogenic organism can be cloned into the gene of a benign carrier organism, usually a virus or bacterium, which can be used as a vaccine without concern that any pathogenic organism are present. The genes or segments of genes that encode the major antigenic determinants of pathogenic organisms can be cloned into expression vectors. A large amount of the product can be harvested, purified, and used as a vaccine. With the last strategy complete, genes produce subunit vaccines and cloned domains of the major antigenic determinants produce peptide vaccines. Peptide vaccines may also be produced by chemical peptide systems synthesis as an alternative to using cloned antigenic proteins or peptides or inoculation. DNA constructs encoding the antigenic protein or peptide may be utilized. These DNA constructs may be delivered directly to animals or humans. That summarizes vaccines here. Let's go to the review questions. Now the review questions. Briefly describe a protocol for developing a vaccine against a toxin-producing bacteria. What factors limit the usefulness of conventional vaccines? As part of your work with an international animal health organization, you are given the task of developing a vaccine against a bovine virus that is the cause of tens of thousands of cattle deaths around the world annually. The viral genome consists of a 10 kb linear piece of single-stranded RNA with a poly A tail that encodes 8 different proteins. The virus does not have a viral envelope, and the major antigenic determinant is the capsid protein viral protein. 2. Outline an experiment strategy to develop a vaccine against this virus. Discuss the development of peptide vaccines that are directed against viruses. What is vaccine virus and how can it be used to produce unique live recombinant vaccines? As an employee of the World Health Organization, you have to decide on the best strategy of eradicating rabies in wild animal populations. Assuming that you must choose between a peptide and a vaccine, a virus-based vaccine, select one type of vaccine and justify your choice. Discuss the advantages of a live recombinant viral vaccine over killed and subunit vaccines. Discuss some of the different strategies that have been used to produce vaccines against cholera. How would you develop a subunit vaccine against HSV? How can bacteria be used as part of a DNA vaccine delivery system? How can vaccine virus be made more sensitive to interferon and explain that? How would you develop a vaccine against S. aureus? Suggest several methods that you could use to deliver DNA for generic immunization to animal cells. What are MIDGE vectors and how can they be used to facilitate genetic immunization? How would you develop a vaccine against human papillomavirus? How would you develop an effective DNA vaccine against dental caries? And how would you improve the traditional vaccine against tuberculosis? That ends the review questions. And now I'm taking you back to the entire chapter from the start for the various illustrations. So you just could keep it as a note there. Right on to the various tables and illustrations and schematic representations. This is the first table on vaccines, 12.1, animal cases in Canada from various diseases before and after the introduction of vaccines against the causative agents of diseases. This is the 4th edition. So just make notes, do research, and update yourself and get prepared for the exams, right? Disease, animal number of cases before vaccine was introduced, number of cases in 2002. You have polio, diphtheria, rubella, mumps, Haemophilus influenza, TB infection, whooping cough, and measles. Take a screenshot of this, right? And make the necessary notes. The next table, human disease agents for which recombinant vaccines are currently being developed. Pathogenic agent, right? You have the pathogenic agent and the disease. So it goes on from the viruses, varicella zoster virus to chickenpox, cytomegalovirus, infection in infants and immunocompromised patients, dengue virus giving hemorrhagic fever, hepatitis A virus, high fever, liver damage, hepatitis B virus, long-term liver damage, herpes simplex virus type 2, genital ulcers, influenza A and B viruses, acute respiratory diseases, Japanese encephalitis, parainfluenza virus, inflammation of the upper respiratory tract, rabies virus, encephalitis, rabies syncytial virus, upper and lower respiratory tract lesions, rotavirus giving acute infantile gastroenteritis, yellow fever virus, lesions of heart, kidney, and liver, human immunodeficiency virus causing AIDS, And bacteria, Vibrio cholerae causing cholera, E. coli enterotoxin strains diarrheal disease, Neisseria gonorrhea giving gonorrhea, Haemophilus influenza giving meningitis and septicemic conditions, Mycobacterium leprae giving leprosy, Neisseria meningitis giving meningitis, Bordetella pertussis giving whooping cough, Shigella strains giving dysentery, Streptococcus gruppe giving scarlet fever, rheumatic fever, throat infection, Streptococcus group B giving sepsis, urogenital tract infection, and Streptococcus pneumoniae giving pneumonia and meningitis, Clostridium tetani tetanus, Mycobacterium tuberculosis, Salmonella enterica cerevisiae typhoid fever, parasites, Onchocerca volvulus given river blindness, Leishmania spp given internal and external lesions, Plasmodium spp given malaria, Schistosoma manzoni given schistosomiasis, Trypanosoma spp giving sleeping sickness, Bushfloria bancrofti causing viruses. Next is the figure 12.1, which is a schematic representation of an animal virus. Viruses generally consist of a relative small nucleic acid genome, 3 to 200 kb of either double or single-stranded DNA or RNA, with a viral protein capsid that is sometimes, depending on the virus, surrounded by a protein-containing viral envelope membrane. This is the diagram of it: envelope proteins, envelope capsid, and nucleic acid. Right? Continuing on to the HSV, which gives a cloned viral D gene, a transfected CHO cell, and a secreted GD protein. Onto a non-protected rat, infected rat, which is protected by injecting the purulent concentrate is injected into the rat. The schematic representation of the development of a subunit vaccine against HSV, the isolated HSV GD protein gene is used to transfect CHO cells. Then the transfected cells are grown in culture and produce GD protein. Mice inoculated with purified GD protein are protected against infection by HSV. Figure 12.3 is a location in the envelope of HSV1 GD with the transmembrane domain, extracellular location of a soluble GD without the transmembrane domain. This shows that NH2 HSV glycoprotein D, exterior membrane, interior with COOH group, transmembrane domain, and figure B is modified HSV glycoprotein D on H2N with the COOH and the exterior and interior membrane. These are the depictions. Please make your notes on that. This is a schematic representation of a subunit vaccine against foot and mouth disease. Antiviral RNA is made into cDNA, which is then digested with restriction enzymes. The DNA fragments are cloned into an expression vector and framed with the gene of the E. coli bacteriophage MS2 replicative protein. The plasmid constructs are used to transform E. coli, and then the stable fusion protein is isolated and used to inoculate animals. The viral RNA, cDNA, that's a complementary DNA. Clone gives cDNA for VP1 fused to MS2 replicates gene, gives immunogenic fusion protein. Figure 12.1, schematic representation of hexameric cholera toxin. The A peptide is shown in blue and B peptide is shown in green. B subunit, A2 peptide, A1 peptide, right? Schematic representation, this is in figure 12.6, schematic representation of the binding of the SARS virus glycosylated spike protein to a cellular outer surface protein receptor. You have the mammalian cell membrane and you have the viral membrane. In between you have the carbohydrate moiety, the receptor there, a spike protein there, right? So take a screenshot, make notes. Figure 12.7 is the main features of a portion of the recombinant plasmid construct used to transfect CHO cells and to produce the fragment of the SARS spike protein that in fact interacts with the host cell receptor. You have a signal peptide, a protein fragment, protease cleave site, spike protein fragment. your clone genes for surface proteins and purify and concentrate surface proteins, right? Take notes on these. The infected mice, both are infected mice. One is not protected. The protected one has injected individual proteins, right? So the development of a vaccine against Staphylococcus aureus. The next is figure 12.9, which is a schematic representation of the virus-like particles assembled from cloned and overproduced L1 proteins from the capsids of 4 different strains of human papillomavirus. These virus-like particles are the constituents of a commercial subunit vaccine against the virus. This is figure 12.10, generalized envelope-bound protein with external epitopes 1 to 5 that may elicit an immune response. This is figure 12.11, structure of a peptide vaccine composed of identical short peptides bound to a carrier protein. You have the carrier protein, linker, and short peptides, right? This is about the malarial infection. You have the mosquitoes, protozoite, infects the liver cells, the merozoites in red blood cells, and then the gametocytes. Infection of an individual with Plasmodium falciparum, a malaria-causing parasite introduced by a mosquito. Take screenshots, make notes. Next is figure 12.13, a schematic representation of Plasmodium falciparum, right? And it is a schematic representation of Plasmodium falciparum, merozoite surface protein 3, and peptides corresponding to portions of the C terminus. The peptides labeled A to F are drawn to scale with the numbers above the whole protein indicating the amino acid numbers, counting from the N terminus. The final peptide is currently being tested in clinical trials for efficacy as a malaria vaccine. This is a former edition, so please update on these notes. Each schematic representation after you read, please update on with the latest. If there's any query that you have or you want me to update from my side, if it's not available to you or you're not able to access or you don't have internet access, something like that, let me know. I'll update that on my video as a separate section, okay? Right. So this is a Plasmodium falciparum merozoite surface protein 3 C terminus, A, B, and C, D, E, F, and final peptide, right? The next is table 12.3, advantages of genetic immunization over conventional vaccines. Cultivation of dangerous agents is not required. Since genetic immunization does not utilize any viral or bacterial strains, there is no chance that an attenuated strain will revert to virulence. Since no organisms are used, attenuated organisms that may cause disease in young or immunocompromised animals are not a problem. Approach is independent of whether the microorganism is difficult to grow or attenuate. Production is inexpensive because protein does not need to be produced or purified. Storage is inexpensive because of the stability of DNA. One plasmid could encode several antigens, vaccines, or several plasmids could be mixed together and administered at the same time. Figure 12.4 shows the survival of DNA immunized mice. Injected mice were immunized with DNA that contained the influenza A virus nucleoprotein gene under the control of the Rose sarcoma virus promoter or an E. coli plasmid. The control mice were injected with plasmid DNA only. The x-axis represents the number of days after the animals were challenged with the live influenza virus. And this is the picture of that survival percent injected control and time details, time in days. This is an illustration of the schematic representation, a schematic representation of the binding of plasmid DNA to the cationic surface of a polymeric microparticle. You have the plasmid DNA, you have the microparticle. Next is figure 12.16, use of non-pathogenic S-flexneri to deliver foreign DNA to mammalian epithelial cells. A strain of Shigella with the deletion mutation of the ASD gene, which encodes the enzyme beta-semialdehyde dehydrogenase, is unable to proliferate and can be used as a live vector. Wild type ASD epithelial cells giving bacterium is pathogenic. Mutant ASD epithelial cells bacterium is not pathogenic. Transform mutant ASD plasmid DNA epithelial cells bacterium is not pathogenic and delivers the plasmid DNA, right? Please make notes. They're very important for your exams. The next representation is a figure 12.17 use of a MIDS vector to produce a capped linear DNA sequence containing the gene of interest, a promoter, an intron, which facilitates expression of the gene of interest and a polyadenylation signal. Excision side, you have the antibiotic resistance gene, the excision side on either side. Then you have the promoter plus intron, gene of interest, polyadenylation signal. Addition of oligonucleotide cap. Make notes. Next is figure 12.18, shows vaccination regimen of rhesus monkeys with DNA containing simian immunodeficiency virus, SIV genes, and vaccine virus carrying the same genes. The first DNA injection and second DNA injection at close to 10 weeks. At 28 plus or around 25 weeks, recombinant vaccine virus booster injection these many weeks from you have mucosal challenge with SIV around 50 plus weeks, right? Here next is a schematic representation of a multi-domain protein encoded by a DNA vaccine. Two domains, the CTLA4 extracellular domain and the immunoglobulin G, IgG, Fc region, are designed to target the other two portions of the molecule, that is, the glucosyltransferase C-terminal domain and the alanine proline-rich peptide to antigen-presenting cells. You have alanine and proline-rich peptide, IgG, Fc region, glucosyltransferase C-terminal domain, CTLA4 extracellular domain, Fc receptor, B-cell protein, antigen-presenting cell. Right? Noted? Make points. The next is figure 12.20, which is a strategy for deleting part of the cholera toxin A, one peptide DNA sequence from a strain of A. cholerae. Note that the tetracycline resistance gene is introduced into the gene for the A1 peptide as part of a transposon. This construct no longer makes A1 peptide but cannot be used as a vaccine because it is possible for the transposon to be excised, and with the result that A1 synthesis and pathogenicity are restored. Have a look at the illustration from the top below. Plasmid A1 peptide DNA sequence given CLA1, XPA1, to a combination of that going to XPA1 linker T4 DNA ligase to the deleted A1 peptide DNA sequence. There's a conjugation which gives chromosomal DNA of V. cholerae. The combination then gives tetracycline resistance gene insert, recombination, chromosomal DNA, plasmid chromosomal DNA, deleted A1 peptide DNA sequence. Right? Note and make points. Next is table 12.4, deleted genes and their functions in the development of attenuated strains of Salmonella spp. The deleted gene and the gene function. Deleted gene would be GAL-E, synthesis of lipoprotein polysaccharide, decreased toxicity from galactose. ARO-A, ARO-C, or ARO-D, synthesis of chorisme, an aromatic amino acid precursor, and a PABA precursor. PABA is involved in the synthesis of iron chelated para-aminobenzoic acid, right? Per-A or per-E, synthesis of purines, ASDs, peptidoglycan, and lysine biosynthesis. Then you have phospho-P and phospho-Q, regulation of acid phosphatases and genes necessary for survival in the macrophage. CYA encodes adenylated cyclase, which is involved in CAM synthesis. Adenylate cyclase. CRP enclosed CAM receptor, regulates expression of proteins involved in transport and breakdown of carbohydrates and amino acids. CDT involved in tissue colonization back to bacterium. DAM encodes DNA methylase. appears to be a master switch with 20 to 40 different virulence genes. STRA enclosed a stress-induced polypeptide result in significantly reduced persistence in human tissues. Right? CMP, cyclic AMP, PABA-SP, aminobenzoic acid. Next is figure 12.21, proliferation of wild type and attenuated L major in mouse macrophages. At time 0, macrophages were infected with the same amount of stationary phase L major. The wild type parasite and the attenuated parasite in the presence of thymidine were able to proliferate, but the attenuated strain did not proliferate in the absence of thymidine in the medium. Hepatitis et al., courtesy to them, National Academy of Science. This is the diagram for that. Wild type, attenuated by thymidine. On the vertical axis, you have parasites per macrophage, horizontal axis, time in culture and days. Note these, make points, and do. Next is figure 12.2. You have immunity to virulent L major induced in BLB by C mice inoculated with attenuated L major at time 0. Mice that were previously inoculated with attenuated L major were challenged with virulent L major, and the sizes of the parasite-induced lesions were measured at various times. Controlled mice were not vaccinated with attenuated L major. Adapted from Tatis et al. To the National Academy of Science. This is the diagram. Vertical axis, you have average lesion size in millimeters, controlled attenuated. The blue is attenuated, where it is control. Horizontal axis shows days after infection. Figure 12.323 is method for the integration into vaccinia-virus of a gene whose protein product, generally a viral antigen, elicits an immunological response. A. Plasmid carrying a cloned expressible antigen gene. B. A double crossover event results in the integration of the antigen gene into vaccine virus DNA. This is the illustration. Plasmid, cloned antigen gene, vaccinia virus thymidine kinase DNA, vaccinia virus promoter, vaccinia virus thymidine kinase DNA, plasmid, the virus vaccinia DNA, the cloned antigen gene, given a vaccinia virus promoter and a vaccinia virus DNA. The next is figure 12.24. You have a portion of a wild-type vaccinia genome that contains the VP37 vaccine. Let's read that again for you. A portion of wild-type vaccinia virus genome that contains the VP37 gene that is responsible for plaque formation in host cells. Portion of a mutant vaccinia virus genome in which the VP3 gene has been replaced by a marker gene C. Portion of a vaccinia virus transfer vector. Left flank and the right flank refer to the DNA sequences that immediately precede and follow the VP37 gene in the wild-type vaccinia virus genome. The native VP3 vaccine promoter is part of the VP37 gene sequence, not shown. MCS is a multiple cloning site with 7 unique restriction enzyme sites. P7.5 is a strong early late vaccinia virus promoter. The target gene is inserted into the multiple cloning site. Subsequently, homologous recombination between the transfer vector C and the genomic DNA of the mutant virus B results in the replacement of the E. coli marker gene with VP37 gene together with a target gene. This is the illustration. Wild-type vaccinia virus genome, left flank, VP37 gene, and right flank. Mutant vaccinia virus genome, left flank, P7.5 marker gene, and right flank. Vaccinia virus transfer vector, left flank, VP37 gene, P7.5 MCS target gene, and right flank. Figure 12.25 is a competitive inhibition of the interferon simulated phosphorylation inhibition of E1IF or 1F2A by protein K3L, which is encoded by vaccinia virus and is nearly identical to a portion of ELF2A. In the presence of interferon, a kinase is activated that phosphorylates ELF2A molecules and thereby prevents them from functioning. B, when vaccinia virus Protein K3L is also present. It is phosphorylated instead of the L2 E1F2A, so the E1F2A remains active. The thickness of the arrows represents the relative flux through each pathway. We have the diagrammatic representation now. Interferon activation, E1F2A kinase phosphorylation, giving the product. Interferon activation kinase plus E1F2A kinase and K3L phosphorylation plus phosphorylation, giving the products here. So where it gives interferon stimulated phosphorylation inhibition, right? This is sensitivity as figure 12.26, sensitivity of wild type K3L plus and mutant K3L negative vaccinavirus to interferon. Mouse L929 cells were pretreated with a mixture of mouse alpha and beta interferon for 24 hours before the virus was added. Plates without interferon had approximately 3 by 10 raised to 7 viral plaques per plate. Adapted from Paolini and Taglia, US patent, courtesy to them, thank you. Look at the figure here, wild type in red, mutant in blue, verticals on the viral plaques, horizontals on the interferons, right? This is figure 12.27. It's a schematic representation of the development of a multi-protein subunit vaccine for tuberculosis. The 6 most abundant secreted proteins from M. tuberculosis are purified from the growth medium and then tested for the ability to induce antibodies in guinea pigs. The immunized animals are subsequently challenged with M. tuberculosis. Growth M. tuberculosis in culture, some protein is secreted into the medium. And then you see the purity secreted proteins, where test individuals' proteins for the ability to induce antibodies and protect animals. This is a figure 12.28, the plasmid construct used to transform BCG to make it a more effective vaccine. The plasmid is isolated from E. coli cells and then introduced into BCG by electroporation, ORI, E. coli origin of replication, ORI-M, Mycobacterium origin of replication, high gene, hygromycin resistance gene and its promoter, P and alpha antigen, the promoter and the coding region of the 30 kilodalton secreted protein. The diagram shows P plus the plasmid is isolated from E. coli, that is P, alpha antigen, ORI-E, M2, HYG, R gene, that is hygromycin resistance gene. Next is figure 12.29, which shows survival of guinea pigs infected challenged with a pathogenic strain of M. tuberculosis. BCG is the traditional live bacterial vaccine strain. Survival, no vaccine in red, BCG plus in green, recombinant BCG in blue, uninfected controls orange. And the horizontal show the weak cycle challenge, verticals about the survival. Figure 12.3 shows using Salmonella as an antigen delivery system and a flagellin antigen fusion protein for presenting the antigen to the host immune system. A flagellin negative strain of Salmonella was transformed with a plasmid containing a synthetic oligonucleotide specifying an epitope. The cholera toxin B subunit inserted into a hypervariable region of Salmonella flagellin. You have the flagellin negative Salmonella transformation of plasmid DNA encoding flagellin cholera toxin B subunit fusion protein, chromosomal DNA. Then you have flagella with cholera toxin B subunit epitope. You have the figure 12.31, which is a schematic representation of an attenuated strain of S. enterica serova, typhi transformed with a plasmid encoding H. pylori, urea subunits A and B into the transcription controller with Salmonella promoted B. The arrow indicates the direction of transcription. P, urea A, urea B. So that's it about the illustrations, the summary, and the review questions. Right? Please make notes and prepare yourself to the best of your capabilities. And get prepared. If you have any doubts or queries or any clarifications, please connect with me on my email or on the WhatsApp here or on the comment section of YouTube of this video. I request that all of you please make the best of notes and prepare yourself with adequate research and updation. If anything is required in that angle too, do let me know. However, please do not copy or reproduce anything for any commercial purposes. Otherwise, everything should be in the best interest of your studies and your future. So thank you, best wishes, have a good future. And I'll get back to you in the next lecture with a new chapter. I'll give you the heading of the new chapter as well. We'll be continuing with the next lecture with synthesis of commercial products by recombinant microorganisms, right? So see you shortly in the next lecture. Thank you. Have a great day. Good Sunday. Bye.